Hello!
Hi there.
Welcome to Microbiology Journal Club. We are not out big about all things small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Then I was a fact checker and editor for pharmaceutical advertisers, and nowadays I'm a member and the president of Biotech Without Borders, a nonprofit based in NYC, dedicated to aid the public with the tools of biotech.
My name is Faz, and I have a PhD in microbiology. I've mostly worked on bacteria, but I've also worked as a research integrity specialist. I'm currently working for a scientific journal.
Every week we meet to talk about, every other week we're meeting to talk about microbiology, and today we're doing an overview of some of the coolest microbiology papers that we've seen in the last 2 weeks.
Yeah, and at some point we'll be selecting a paper to do an in-depth deep dive into. So message us at MicroTWJC about what paper you'd want us to look into, and we'll see if something has caught if you we'll dig deep into it for you.
Absolutely. You can follow along with any of the papers that we discuss or things that we're thinking about discussing in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet us at MicroTWJC. And boy, do we have a show for you. First, we look at the new Omicron subvariants, which are taking over the world. What else do we do? Yes, we delve into SARS-CoV-2 pathogenesis, what it's doing in the lung, and looking into how it can spread and how it alters cells. Then we go all the way back in time to the origins of life and photosynthesis, and seeing how scientists attempt to resurrect ancient versions of some of the most important enzymes in the world. Not only that, we look at about how our microbiomes are spreading to other animals, and whether snail entrails can tell tales about the microbiome. Also, we look at how some viruses can make you smell good to mosquitoes, and finally, we look at how funguses can protect bacteria from oxygen.
Mm-hmm.
And
So yeah, as is our regular program, we often cover a bunch of SARS-CoV-2 papers, ongoing pandemic, and yeah, we talked a little bit about BA.4 and BA.5, but we do have a couple more to talk about today. This first one is antibody evasion by SARS-CoV-2 Omicron subvariants BA.2.121, BA.4, and BA.5.
yeah, these are variants have. So I think the BA.4 and BA.5 originated in South Africa. BA.2.12.1 came from the US, is as I understand it. At
Yep,
least I know that that's
New
the one
York
you've
City, maybe.
accused of giving you COVID.
Yes, yeah, I think that's the one that I caught. Just based on the time and like the looking at like the CDC like prevalence rates at the period that I got COVID, I think that was the one that caught me. I wasn't being careful. I was unmasked eating food, so like it's my fault.
I mean, I think I've just been very lucky at being able to dodge COVID. For the thing is, now I know that the moment I say that I've dodged COVID on this podcast means that I'll probably get it very soon because COVID will know and get wrathful and try and find me. So
Yeah,
I have to be very careful about. Oh well, it's just unlucky, I guess.
right.
Maybe
Well, we've talked
need to go into
before.
my bunker now to avoid COVID.
I feel like we've talked about that like Swiss cheese method of avoiding COVID a lot on this podcast. Just being that everything's an imperfect barrier, right? So the more imperfect barriers you put between you and the virus, the better the chance you have of dodging it. In a lot of places, I feel like the mask barrier is being eroded. People's willingness to wear those things and stuff is going on. Yeah, and these this BA.4 and BA.5 variant we've talked about before on the stream, saying that it is evading, based on neutralizing ability of pre-existing serum, it seems like it's evading it, and this paper is giving us more data along those same lines.
Yeah, so like so I think this first figure shows like how BA 4 and 5 and BA 2.12.1 are kind of have been expanding across the world.
Mm-hmm.
So you can see like where they're so like globally they're taking up the BA 2 is still not gone. I mean, still like all Omicron is taking
Yeah,
over,
it's been a very
but
effective lineage, right? There's been a lot of variants just within it that have been, yeah, that that are the ones that are circulating these days.
yeah, you can see the difference in the US and South Africa, where South Africa is mostly the BA.4.5 and USA is mostly BA 2.12.1.
And this is
And UK, it's a mix of both.
June, early June is the last time point they have here. So like now in late or mid-July, I've seen the CDC for the USA. The proportion has definitely changed. There's a lot more BA4 and 5. I think even here they're not delineating between 4 and 5, but I think I saw in the news that 5 is even higher than 4, something like that.
Yeah, yeah. 5 is the new variant, because we've been through this with like all the different new variants.
Oh my gosh,
right?
yeah.
I think at least 3 times and now is a traditional looking at the neutralization assays to see how this variant is messing with our immune system.
And of course, the serum that they're able to collect to look at this, the ability of the serum to neutralize is serum that comes from vaccinated individuals, right? Serum that has come from people that have are convalescent for a variety of the different earlier variants. They have a whole smorgasbord of pairwise interactions that we can interrogate.
Yeah, and like they also had the traditional looking which monoclonal antibodies work and don't work in order to determine what the mutations are doing, which they showed a bit earlier. But basically, I think so. They've got a couple of pairwise comparisons. I think they kind of show that. I'm trying to interpret this on the fly, but I think that mostly like Omicron infections are the serum from BA1 breakthroughs and BA2 breakthroughs are better at neutralizing, I think, than most of the strains here, right?
Yeah. I mean, they're not. The last two in A here, right? The last two panels, they have the serum from these 2 different types of breakthroughs. It's the least effective at neutralizing Omicron, but it actually is the most effective at neutralizing the ancestral strain.
Yeah.
Yeah. Which is, I guess, like there was this thought, right, that Omicron isn't giving us, maybe it's the nature of Omicron itself, right, isn't giving us like a wide breadth of neutralization capability for like the other Omicron strains.
Yeah, it's
BA1. I guess it's pretty good against BA1. But still less than it's I'm just looking at like the cross between it with BA2 convalescent serum, like it is a little bit worse with BA1, but the best for BA2, not as great for BA4 and 5.
Yeah, I mean, I think like the BA2 is the one that has the best neutralization overall. I'm just trying to look through this to try and figure out, because there is also a decline effect that happens where the time since you get boosted, that might have. So I'm wondering whether that might be having. Might not. I
don't think they didn't segregate by that in any figure as far as I can see.
Yeah, I mean, I think they took them at specific times. So these aren't like things that these are serum that are on file rather than. So maybe that isn't as much of a factor. But the thing that was quite interesting is like this 3-dimensional plot they created about kind of to illustrate of neutralization between the different sera.
Well, and it seems like all the Omicron variants are spread out along like a big. That's how I'm seeing that, if that's the interpretation.
Well, yeah, the thing about 3D graphs like this is they're really hard to interpret, which is why you get a lot of things like with the UMAP kind of diagrams, which kind of flatten out those kind of extra in order so that we can see the relatedness more. This is very. I mean, it looks like BA4-5 and BA1 are very far apart. But yeah, there is.
They're like as far from BA2 as D614 as the ancestral D614G is.
Yeah, that's basically the impression I'm getting from this.
Yeah, and again, like in the papers that we looked at before, where they were doing this in cell, right? We're looking at cell culture and other serum. I feel like we've seen this data before. This is like just another packaging up, and maybe we didn't see so much on BA 2.12. But I remember seeing this from BA4 and 5, like in a preprint when that was first emerging in South Africa, right? We're well in the know that this one is evading immunity, and it's borne out by the rates, right, increasing. I think that's like, I don't know if that's like comforting, but like, you know, that's like, we can feel confident that like that's probably what's driving this.
Yeah, well, the new variant emerges. It's usually not whether it evades immunity, but how it evades immunity. That's a question.
Right, right.
And looking at like what antibody, like monoclonal antibodies, still work, and
And,
I
oh,
think
and I think
which returns us to the
I was going to say, especially from the idea of like, what is it replacing, right? Like, what's the cross protection from the variant that is moving out? And for most places, that's going to be BA.2, I think. And so the idea is that if BA.4 and 5 is quite different, it means that we would expect to see like an increase in cases if other precautions aren't being taken, I guess.
Yeah. So I've got another short like SARS-CoV-2 paper lined up. Is there anything else we want to?
Yeah, I'm good. I'm good with this one.
Okay. So the next paper is titled SARS-CoV-2 Omicron BA.5: Evolving Tropism and Evasion of Potent Humoral Responses and Resistance to Clinical Immunotherapeutics Relative to Viral Variants of Concern. Now, this paper, this does again the dance of like the neutralization which we saw in the previous paper, although this is slightly less detailed than that. But what separates this paper was that they looked at the tropism. So one thing that
the type of cells that it infects.
Yeah, the cells it infects, and also like how it infects. Because the Omicron thing that was very strange was that it was less good at fusing. It was the kind of furin cleavage site for the Omicron strains just wasn't as good. So the idea was it was actually going into the cells in a different way.
Yeah.
However, for this new strain for BA.5, this paper, they test out the ability for it to infect cells which express ACE2 TMPRSS2 compared to like the original BA.2 variant. And what they effectively find was that this appears to be cleaving furin again. So instead of this has gone back to the basics, back to that killer app of the original SARS-CoV-2 virus, of it being able to infect cells and cause fusion.
Yeah, because I think we saw something earlier where it was like Omicron might have been using, or like the BA.1, BA.2 Omicron was using like the clathrin, or some sort of endocytic pathway where like it induced like a vesicle to be formed and then it fused in the vesicle time. But yeah, I think the finding here seems to be that it might be just back to fusing at the surface of the cell.
Yeah. So what they kind of found was that this BA.5 strain, it can cleave TMPRSS2, and you can block it with nafamostat, which is one of the TMPRSS2 like blocking things. So again, this and I think they kind of like tie it into it being kind of more invasive, because the thing about Omicron was like we thought that because it used the endocytic pathway, it sticks to the upper respiratory tract a lot more, whereas using the TMPRSS2, that allows it to infect a lot more areas and be slightly more invasive. So that cells it infects.
Yeah, so actually I do think that's interesting, because like we're always, I think the most difficult thing that we've had in explaining is just like where, like what what are the advantages of each variant, right? And there was the thought that the upper respiratory infection was making it more transmissible, but then here we have maybe a combination, right? It's finding some way where it can still be very transmissible, but still also get to a wider range of cells across the entire respiratory tract.
Yeah, I mean, sometimes not every difference is adaptation. Sometimes, I guess, in that case, Omicron benefited because it was so different from all the other SARS-CoV-2 variants that the immune system basically treated it almost as a new infection. So while people were still protected from severe disease based on the vaccines, that you could still transmit it in the same way as the original. And with these BA.4 and 5, because you've still got that unfamiliarity, it means that adding this in can kind of bump up its infectiousness even more.
Yeah. Yeah, I mean, I think we still think that the vaccines are protecting against severe disease. And I think that that's also borne out by looking at sort of the epidemiology of it, where like there isn't that much. But then, of course, okay, we always say this: the more people that get infected, right, the more potential it is to hit the vulnerable individuals that are going to be that their outcome will be serious disease from the virus.
Yeah, and I think there is now a much stronger argument for creating an Omicron-centered vaccine, because I do feel like the chances of another variant appearing and taking over seem to be a lot lower.
Yeah, it seems like Omicron is the most successful because of all the variants within it, right? That this has been the most successful branch. And then based on based on the thing that we saw, the data we saw in the last one, and sort of some of the understanding that we had, like there doesn't seem to be like this cross, like there might be advantages to finding something common across all the Omicrons that would be able to produce like a vaccination response, instead of relying on the.
I do want to bring back the previous paper where like these, like say the BA.2 breakthrough infections, where you still get them neutralizing the original like recipe strain.
Yeah.
I think that's some.
Well, because I think we talked about it on stream before too, that there was a thought that maybe if we made variant-specific vaccines, then we would be losing cross protection, right, to things outside of that specific clade. But yeah, as you say, if breakthrough infections were giving us cross protection to the ancestral, then maybe that's not so much of a concern.
So yeah, I think the thing I'm thinking of is like as a booster, it would be good. Maybe not as the original kind of vaccine because of this thing called immunoimprinting, where the immune system will react like specifically to the original version.
Yeah.
Well, will kind of imprint on the first response and tailor its responses to that. But that then like any subsequent response will be like another version of that same original response.
Yeah.
So
yeah, and I mean, it gets really complicated, right? Because I think also like thinking about what that effect will actually be if we do get a unique Omicron vaccination, there's the question of like some people have had, like everyone has had a different infection history from right, from both an infection history based on if they got the vaccine and then what sort of strain they had their breakthrough infection from. I don't know, it's kind of interesting. I mean, I guess theoretically, the breakthrough infection serum is sort of like if you had that original vaccine, right, and you're getting boosted by this other antigen. But of course, infection is not vaccination. It's like it tickles a whole bunch of different immune responses that could drive towards epitopes that aren't the optimized ones for driving off broad infection. I guess I don't really know, actually. This is quite confusing. I've heard that in the news that people are saying that maybe we should have a Omicron-specific vaccine. I'm just curious what the proof of evidence would be what would we want to see in a clinical trial there that would make us feel like, oh yeah, like this is really great. Like how will they have to enroll people, I guess? I'm thinking about the exclusion criteria. Are they going to do it broad, like you can come with any infectious background, or are they going to be like narrow about that?
I think that they probably want. I mean, what I'd be looking for a study would be looking at the effect of an Omicron booster, because most people now have had that original SARS-CoV-2 vaccine.
Right. Oh yeah, sorry. I've been saying vaccine, but I have been thinking like it's a specific booster. Yeah.
Yeah. But I mean, that's one.
But I mean, I guess what I'm saying, to show efficacy of that booster, is it just enough? I guess maybe it's good at people will just accept that it's safe and so they'll use it. But like, how do you show efficacy of such a booster in the current environment? It seems complicated. And like I guess I'm not familiar enough with like what it looks like to get boosters through. What it is to like have a formulation of a booster change and then get that through the approval process. Because I don't know if we really see that, right? Like we don't see the regulatory system. I can't think of an example actually where that happens, right? Where it's like, oh, we made a booster of like something slightly different, and now we want to give this to a lot of different people. I mean, the pandemic I think has highlighted some of the new ground that we're experiencing during the pandemic, like different problems that the sort of like the larger healthcare system hasn't had to think about before, but like I guess by necessity is being pushed towards thinking.
I mean, I feel like we almost have an example of that where because I got like my first vaccine was the AstraZeneca vaccine, and I got boosted with the Pfizer vaccine, and I think that that's something that there was a conscious choice to switch over between those, and I feel like that's almost like the first step in deciding like what's going to be good for a booster or not, and using that kind of similar framework.
I guess but like the data that came from approving that cross, right, like the cross boosting thing, that data came from seeing like a sort of a typical clinical trial where it was like a naive population that didn't have anything, that some of them got vaccines, and then you could see the efficacy, right? And then they just like kind of made, in their mind, they were just like, well, it wasn't, I think there was like a necessity element there where it was just like, we need to give people their second dose, right? Like manufacturing things have to happen. Yeah, I don't know. I feel like I'm actually, maybe I should be listening more to the news about that type of thing. Like what are regulators saying about the process to get a booster through, like a specific Omicron booster through?
Anyway, speaking of, well, not speaking of, but I'm segueing into the next paper.
Oh, yeah. Let's go to the next one.
Yeah, next.
Lots of like SARS-CoV-2 navel gazing.
Yeah. Oh, it's been 2 years.
Yeah, this one actually I'm quite excited about. Bronchial epithelial from adults and children, SARS-CoV-2 spread via syncytia formation and type 3 interferon infectivity restriction. So again, going back to this thought of like, how have we been characterizing the different variants of SARS-CoV-2, often through the spike protein and often through like assays saying like, oh, it's pseudotype viruses like get inhibited by this by neutralizing antibodies, but then also these cells are better at infecting certain cell types or something like this, right? They look at the dependency, like what we just talked about, the dependency on whether they fuse at the initial membrane or whether they get endocytosed. But then we've also seen this syncytia assay where they say, oh, in a cell culture, how many syncytia form, right? So how many blobs of cells get created because the fusion protein on the outside is just like having the cells fuse. But like it's always the question in infectious disease is like, we have these interesting assays, these phenotypes that we see sort of abstract from the overall infection. What is the role actually in infection itself? And so in this paper, they have this interesting epithelial model. It's like an explant. It's kind of like a cross between an explant model and an in vitro like cell culture, but essentially it's the cells from individual patients are cultured into a 3D cell culture, and then they can infect that with SARS-CoV-2. So it's kind of like infecting, right? Like there's a distinct genetic identity to the different things that they infect, and they're human. So like that's why I find it a quite exciting piece of research, because I mean, it's almost like the next best thing besides infecting humans, right? Like in terms of like the quality of data, not like the ethical considerations. I mean, this is the best thing in terms of gathering this data.
Yeah, it's quite interesting how they get quite close to how like it's almost getting a lung in the petri dish, which is quite. They have this air-liquid interface where they put like the cells on, and the cells differentiate into like almost a lung epithelium where you've got like different layers forming.
Yeah, and they even validate those layers by looking at how well the cilia move and how well they move mucus. So like these layers have like a functional element to them where it's like demonstrating that this is like the tissue that we're sampling from the human. And so anyways, they have different individuals and they can infect with SARS-CoV-2. And they can see that these syncytia form on the apical surface, on the top of the on the top of the infected cell culture. And I think,
No, sorry. I was thinking this, I'm trying to find the right things to demonstrate what you were talking about.
Like an image of the syncytia on the surface, right?
Yeah, right. Because I think they've got like
yeah, I'm not sure what, I think in figure 1A, yeah, you can see it because like there's like these like foci, but maybe there's like a better zoomed in version. Oh yeah, no, in figure 2D has a 3D scan sort of what's going on.
You figured to be
2D, 2D and 2F, yeah. 2D and 2F.
right.
You can sort of see the surface. They're able to stain for the N, right? N is the nucleocapsid protein of the SARS-CoV-2, and then they also are staining for some cell markers. And you can see, like, especially in F, I think, has like a really good view. Like, this is the apical surface, right? And you can see that some cells are smaller or bigger. Like, some of them are really giant, right? And those are the syncytia. Essentially, like a bunch of little cells that a cell that got infected fused with neighbors and became a really big cell that's full of viral replication. Actually, that's what the interesting thing they find out is that when they wash off the syncytia and when they make washes of their air-liquid interface and then they fractionate it to see what part is infectious, like it's the part that has all the big syncytia cells have the most virus associated with them.
And that's a really, really interesting finding. I think the part of it is that I think they find that syncytia almost like pop out of the epithelium as well, like that they bulge and are more easily washed off, and that they themselves
Yep.
are infectious, like kind of like it's. I mean, it makes sense for the virus because it can use the whole cell as a defense against the immune system, because the immune system will see these syncytia going on and assume that, oh, maybe they're normal cells, and then it can just pop out almost like a virus cluster bomb, just wandering around like a reverse virus Katamari, where it just dumps virus everywhere.
Yeah. Yeah, I think it's also kind of like, it makes sense from like a success, like evolution and success sort of thought process, where it's like a syncytia is essentially a very successful site of infection. And so like it makes sense that the successful sites are the sites that are then distributing virus all over the place. And every cell that gets infected has a chance of becoming that big syncytia that then has this huge payout. And this is sort of infection in the context of tissue, which I think is helpful to think about. It's not just this surface is what the virus essentially does see, right? When we have particles enter our nasopharynx or whatever.
Yeah, I think they even like delve into like how the virus specifically goes out at the apical surface, not the basal one. So it doesn't like go deep. In fact, when it does go deep, that's when it forms syncytia. Because I think they looked at these cells, and there's like cells in the bottom layer, cells on top of it, and they found that syncytia like have the markers of both.
Yeah, they're fusions not just of the stuff on the surface, but fusions of stuff from the bottom. At least that's their ongoing hypothesis. They just have some cell staining stuff to go on. But yeah, I think that that's a really interesting thing as well. Like, they're not looking to punch through the barrier. I mean, why would they anyways? They want to spread through respiratory droplets. But in going deeper, they do find cells they can merge with. Yeah, that, like, who knows what these syncytia are I actually don't know how the immune system would see such a cell, right? You would hope that they would do apoptosis and just stop going, but actually that's a good segue into our next one. But I do want to say one more thing about this, is that because they have genetic variation, they have like cell cultures from kids and adults, right? And these are presumably like individuals in a way, de-identified individuals. They actually see variation in the infectious like outcome of their experiments, and they find this really odd thing. Well, first of all, the kids' bronchial epithelium that they reconstitute are fairly resistant, and that sort of makes sense, right, based on what we know of like kids being infected and like what their viral burden looks like. But they also find some adults that have like a similar profile where like the viral burden isn't so high in the cell culture. And then that's kind of cool because they're able to pinpoint they're able to say that the difference between these adult cells and the ones that are more permissive is like some interferon expression pathway.
Yeah,
And so they can make
lambda. I think they pick out.
Yeah, interferon lambda, which is actually not. I actually don't know anything about interferon lambda.
One of those rare ones where it doesn't come up often, so I never had to really learn about it for my exams. So it's one of the ones that So you learn about alpha, gamma, beta, and actually I don't think you learn about. But anyway, But yeah, another cool thing they do is they use CRISPR to knock out some of the genes in these. So with primary cells, you take them from a human and they grew them out, and then they use CRISPR to knock out the function of interferon lambda to see the effect it has.
Yeah. Yeah. And so it's a very nice control in some ways, right? Like to verify this observation. So they made the observation with the variants that already exists within their samples, right? They have different adults, and then they go in and say, okay, well, let's experimentally then create the lesion that we think is responsible and see if it recapitulates the phenotype, and it does. So yeah, I think that's like just a really cool way that they approach this.
I mean, the one thing the fact they have lots of variation is something that is very good. Part of me, so the thing I'm looking at is G&H, where they do like a knockout in like the same adults, but they find that the variation of the knockout of interferon can be quite different for different ones, kind of indicating that this is
yeah.
Which I love, because it does mess up a little bit their story, but also indicates that what you'd expect, the immune system is much more complicated than just one protein.
Right.
This kind of demonstrates that interferon is having an effect. So it can be an important effect for some people, but it's not all there is.
Yeah, it's not like if we had some way of giving this interferon to everybody, everything would be better, right? Like there's because again, like interferon, like it's a signaling molecule in the immune system. So like, yeah, you can like start sending signals to the immune system, but if you have other variations in your immune system, you may respond to that signal differently. Yeah, and so that's like the complexity of the whole kit and caboodle. But yeah, I think just the information they're able to gather from this particular model system seems really interesting and stuff that I hadn't heard before, right? It gives us some significance. I was really excited to hear about a potential significance of syncytia in an experimental model.
Yeah, completely. Yeah, so I guess
one more SARS-CoV-2 paper.
Yeah, one more SARS-CoV-2 paper, and then it's not over. It's still going to be happening, but we won't be talking about it as much. So that is the role of NSP6 in the biogenesis of the SARS-CoV-2 replication.
So yeah, just like as we said, or the place where it could have segued from the last one, but I want to go, there was sort of more details to pick up, was like the syncytia are these giant virus factories, and inside of the cell, like there's all these rearrangements going on. I think we saw actually very early, right? We saw like the pore. We covered a paper on the pore in membrane vesicles that's made by SARS-CoV-2. So like they essentially create like special locations. Here they're calling them double membrane vesicles. It's the replication organelle, where there's some shelter from the intracellular sensing pathways. And so this paper is, the role of NSP is looking at the role of some other proteins in creating this space for viral replication.
Yeah, NSPs are like the proteins we don't hear. We all hear about the spike, but NSPs are the help the cells, like once it's in the cell, to start creating viruses. And they did here, I think they looked at a couple of different NSPs to see which ones were important for creating virus factories in creating that replication organelle. And
I'll say the most important NSP is like polymerase, right? Like that's a non-structural protein. But like as you go down the list, like I think NSP1 is probably polymerase. As you go down that list, you get to increasingly more obscure, like small proteins that somehow signal to our cells and mess up the interactions that can take place.
Yeah, and this paper kind of focuses on the creation of this replication organelle. And I think they focus in on how it's derived from the endoplasmic reticulum, which is this kind of protein factory that surrounds the nucleus. And what they found is they looked at the structures of the NSP6, and they kind of figured out that it binds to an area on the endoplasmic reticulum. You get to see a lot of like immunoelectron microscopy to see that.
Yeah, they find like a very interesting structure. I hadn't really thought, I don't even actually know after reading or after skimming this a little bit, what exactly is the zipper. Like it seems like it's like some sort of like extended, like thin, maybe it's like single membrane. It kind of like comes from the endoplasmic reticulum and it touches, and then it connects the endoplasmic reticulum to the replication vesicle. That's what I'm understanding.
Yeah, that's what I kind of feel like, that the replication, it kind of like pinches off a area of the endoplasmic reticulum and
Mm-hmm.
kind of creates an area for like almost like creates an area for the replication organelle to assemble.
Yeah.
Let me see if I can.
Because of course, like the virus is making these replication organelles from the components of our own cells, and so yeah, I guess the zipper is maybe the transitionary state of the membrane from the endoplasmic reticulum going into the replication vesicle. Like something that might help folks understand that concept is that inside of our cells, it's like everything's membranes in terms of like being able to compartmentalize places. And if in some ways all the compartments are just evolutions of pieces of membrane, either from the outside or from the inside. And so in the process of trying to make a new membraned place to replicate virus, you would necessarily have to interface with the existing stores of membranes. Yeah, and here it seems like NSP6 is governing the ability to take membrane away from the endoplasmic reticulum.
Yeah. So I think in this like image, it kind of shows like two areas of membrane coming together, and they kind of, so the black arrow, they kind of like indicate that maybe that's caused by NSP6, and we've got these fancy like 3D models where they find that there are two other proteins, NSP4 and NSP5, which interact with outer membrane vesicles. Well, not outer membrane, no, double membrane vesicles are kind of just naturally produced within the cell, and those can interface with, I think, with NSP6 in order to create these like networks for the replication organelle. And that was my understanding from my kind of read of the paper.
I think it'd be thought of like the virus is like needing to get membrane from somewhere, and so it's harvesting from these different sources. And NSP6 specifically has, so they have like a mutant of NSP6 that like is missing 3 amino acids, and then that one doesn't do this job as well. So like they're sort of proposing like that it's like an anchoring, right? Something about NSP6 is like it sits in the membrane and it somehow associates with things to bring them together in a way.
Yeah, it's almost like an organizer for how the replication organelle will eventually develop.
Right.
So they sort, because we don't know the full structure, but they kind of theorize that it like kind of oligomerizes, so it like sticks with itself in order to get into the membrane, and then it's kind of uses that reaction to zipper like different parts of the endoplasmic reticulum together to create these tubes, and then links up with these double membranes with NSP3 and NSP4. I misspoke earlier when I said 4 and 5. So like they found like the kind of NSP3 and NSP4 kind of like are homogeneously distributed through the cytoplasm until you add in NSP6, when that kind of pulls everything together into these double membrane vesicles.
Yeah, so it's a very in-depth, like this process is very complex and it's not fully mapped out for SARS-CoV-2, and this is maybe this is like in the weeds, really. It's in the thick of it, right? Like in terms of mapping out like that process, figuring out what proteins are involved, what do they need on the host side, what do they need on the virus side to make it happen.
Yeah, this is very much in the kind of school of finding out how viruses like basically cannibalize all the things inside the host and use it for what they want. And that part is like quite fascinating for me personally, because because of the biochemical aspects, the fact that you've got this kind of invader in the cell and it needs things, it needs to consume protein, it needs to consume lipids. How does it do that? And this kind of like shows like lipid drop and other things to kind of pull together to create this entire like basically a virus organ.
Yeah, I mean it is a viral organelle. I think that's that is how people think of it as well. It's a parasitic parasitic organelle that has all these abilities.
Yeah, again, they're like kind of that whole viruses are technically like dead. Well, they're not alive unless they're inside a living cell. In which case, that's when they have their metabolism and everything else.
Right. It's a parasitic metabolism, totally dependent. And I think it's like really cool that they don't have. It's only like a few proteins that are required to sort of like create this subsidiary, this alternative organelle, rearrange everything. Just a few proteins does it.
Yeah, I mean, the viruses are the original like biotech like entrepreneurs.
Oh yeah, they're disrupting the system.
Yeah.
They're moving fast and they're breaking things.
They're breaking rules, and importantly, they're taking more resources than they need from the society. They're not cooperating. They're
oh my gosh.
Oh.
All right. So that's the end of our SARS-CoV-2 coverage. Pandemic's still going on. This most recent variant is it's out there and it's increasing. So take care of yourselves. But scientists are also figuring out all these really cool mechanistic things about how it works. Don't know whether or not we're building towards trying to get therapeutics now or what the information is, but of course the microbiology is fascinating and we're interested in learning more about these life cycles just because it's cool stuff.
Yeah, and that brings us to our next paper, which is earliest photic zone niches probed by ancestral microbial rhodopsins. So this is an interesting paper because we don't usually like sequence gazy papers, but I saw this one and it really like caught my eye because what they're essentially trying to do is trying to figure out like what an ancient protein looked like based on all of its descendants. So it's a very like kind of molecular paleontology kind of a paper, which is kind of interesting to me. And the idea is rhodopsins. So rhodopsins are they convert light into energy. It's very similar to like how chlorophyll does, but we're very familiar with them because in the human eye we have rhodopsins, but they're also found in microbes, and I think they're arguably, I mean, some most like most widespread form of photosynthesis out there because there are a couple of really tiny microbes that live on the ocean surface that use microbial rhodopsins synthesis.
Yeah, I think that they can be like attached to pumps. So like some photons hit something and then that allows them to move some ions against their gradient.
They're able to like effectively changing shape when light hits them allows it to pump protons through, and it's a lot more simplistic than chlorophyll and other
because
forms of photosynthesis.
In photosynthesis there's like a huge like chain of like proteins that like takes an electron and like reduces it down and like absorbing small amounts of energy from each time. Like these are more like one-stop shops. I think like light hits them, something happens, and then they do something that's slightly beneficial.
Yeah, and so like there has been this argument which came first, chlorophyll or rhodopsins, and there isn't really much evidence. It's very controversial because literally bacteria don't form fossils and neither do chemical reactions. So it's very hard to figure this out. So this paper's trying to get back and kind of roll the like whether that structure of these ancient microbial rhodopsins can tell us anything about those sorts of questions.
Right. So if you roll us to figure 1, you can see one of these. Like there's tons of rhodopsins, right? Bacteria. There's tons of bacteria. There's tons of existing rhodopsins, and you've probably seen these before, right? We talk about them often when we're looking at microbiota. They make phylogenies, right, where they look at the relatedness of something. But these aren't bacteria. These are just like the protein sequences. So this is a phylogeny of protein sequences where they're saying like this is the most likely. This is the most the way of representing all the protein sequences that we know existing, how they're related, with like the fewest amount of changes. And it all goes back, right? Because the assumption here is that they're all evolving. It's like a radiation, right? Like diversity is coming from some ancient ancestors. And so each of these nodes, at every node here is like, there's an ancestral sequence. And like, we don't have an example of it. Like, it's not like we found these sequences in nature. They're just inferred from saying that if all the ends of the trees, and this is the most like parsimonious, the lowest amount of like the most, the simplest way of representing all the relatedness, then these are the implied ancestors that must underpin that variation.
Yeah, I don't know if you've seen those Pokemon memes where they try to like figure out what the ancestral Pokemon looked like, where they kind of reduce. It's kind of like that, but if you don't know what that means, I'm talking crazy at you, and I'm very sorry.
Yeah, what's more? I don't know what's more likely. If folks are watching, would you know like phylogenies from Pokemon or phylogenies from looking at sequence?
I don't know. They're both like deep dive wise. So like you have to be like both like an intense Pokemon fan and an intense microbiology fan. In which case, I don't need to explain either of these things to you.
Yeah, but I think from the like looking at a tree though, that's sort of like understandable that like the nodes that are like older on the tree, more to the left, right, are like the ancestors, and like those. It's not that they have that data, right? That data is just like an inference from looking at things, yeah, from that are from the present. But like really strangely in this paper, or like I guess this is the cool part of this paper, is that then they have another algorithm to lay on top of this, where they can analyze the ancestral sequences and sort of try to predict what wavelength of light is going to be used by them.
Yeah, that's like you can even see it. They try to indicate it on the phylogenies, like what wavelengths of light the current strains like absorb into. So we currently see them absorbing green and yellow, which is again quite different from chlorophyll because green is the one thing that chlorophyll reflects.
Yeah.
So we'll talk about it a bit more, but yeah, what they do is they got this algorithm that can predict, and they show. I mean, and you can kind of see how it goes with it because like each of these kind of roots have a color that is a mix of all the other colors that came before, and from that they kind of try and work it back to figure out what the original version like absorbed, and they go, oh, it absorbed green light, which makes sense because green light is the predominant wavelength that is put out by the sun. So it would make sense for something to evolve towards that way. So I mean, the other phylogenies that didn't survive, I imagine, would have absorbed other colors. So if there was like a phylogeny that absorbed blue, like right at the bottom, that didn't get any descendants. But the winner would have been a green phylogeny, I guess. Yeah.
Yeah, it's like a strange, it's like some sort of machine learning was used here in order to make the prediction of what wavelength it would be. Because like this is one of those hard problems in biology, like sequences, right? Like sequence of structure, and even then functionality after that. Like it's not an easy thing to predict. And I think they're using something about the relatedness to help, to some information about the relatedness also to help. Like it does take into account like the wavelengths that are coming out, like the extant wavelengths that are being used.
Yeah. And they talk a bit about like how there are different like motifs out there that so there's like a DTD, DTE motifs which are kind of located within the which indicate what kind of thing that rhodopsin will be pumping. So so there are some rhodopsins out there that pump chloride ions, and they're called halorhodopsins. They tend to be more yellowy, but they have a TSA motif, and the other rhodopsins that work in the kind of the work with pumping hydrogen ions, they tend to be DTD, DTE, and DTF. Yeah.
roll us to like I don't know, figure 3 maybe, like so in figure 3 like they're actually showing like this is the structure that they have, like because there's structures of like existing ones, and so they just like scaffold on, right, like their ancestral sequences onto these known scaffolds and look at like, oh, where would the changes be and like how might that affect something. Yeah, it's all sequence gazing here, right? It's all through the power of seeing what exists and looking at the relationships and sort of inferring ancestral relationships from what exists now.
Yeah, and essentially what they kind of find is that so they a lot of it is also like kind of there's a lot of like talking about this. There's a lot of like
Discussion.
this is almost partly a review paper as well, and the way they talk about things, they do refer a lot to previous research. It does support an old, a much more like mainstream theory that these photorhodopsins occupied a niche that other green plants didn't. So while that and potentially like there's almost this idea that green plants are green because the main rhodopsins that were out there were all in this kind of absorbing kind of purple. I think the idea of purple seas.
Yeah.
And also the idea that also like the depth of where, because green light like travels a bit further down, and if and back at this we're talking about like these rhodopsins were expecting Earth, so really old, no oxygen in the atmosphere, no ozone layer, so there's a lot of UV radiation. So they inferred kind of based on the light they absorbed, where in the water column they'd have been able to survive, because that's there's like a sweet spot for green light where you get most at the surface, but at that surface you also get killed by UV light. So there's a sweet spot where the bacteria live where they're not getting killed by UV light, but also absorb green light.
Yeah, it's wild to me that like maybe this is like the power of scholarship in my mind. It's like you don't need that much, or I mean they have a lot of information, but like it's just about like sort of like the way they systematically went in and like started to tie in all these different theories together to say like, yeah, like we're by making this estimation, I guess that's what it is. This paper impresses me in the way that by making estimations, it sort of brings us closer to potentially understanding what might have happened in the past, right? This estimation, who knows like how great it is, but just the fact that they were able to construct it gives them a way of interpreting observations and theories that they weren't able to previously think about. Or that were previously thought about. They're supporting, actually, right? You said they're supporting something that already exists.
So it's very interesting, but again, it is very sequence gazing. And they always thought like that they have the sequences. One, they just create the proteins, which brings us to the next paper where they actually do that.
Yeah, so exactly the same beginning to the question, just saying like we have all these existing Rubisco enzymes. So that's the enzyme that does carbon fixation. So resurrected Rubisco suggests uniform carbon isotope signatures over geologic time. Yeah, so they.
I want to stay on Rubisco for a little bit because it's like apparently according to like my A-level teacher was the most enzyme on in the whole planet, like very important for the carbon cycle. So there's this thing called the Calvin cycle where like you have photosynthesis that generates energy, and then that energy is pushed into the Calvin cycle, which actually locks in the carbon, creates all the sugars and all the carbon that allow us to be carbon-based life forms. So yeah,
like we have the whatever ancient photosynthesis individuals existed, or like populations existed, we have them to thank for all the carbon biomass. Because like there weren't complex, this is like we're thinking about like the beginning of life, right? Everything, we don't think of like, oh, there were like these huge carbohydrates like floating around. It was like the chemistry was simpler then, and Rubisco being able to attach carbons to carbons is sort of the gateway to all sorts of molecules that we have.
Yeah, it's the reason why we have oil and Russell Brand. So yeah, everything.
Yeah, so in this paper, they do the exact same thing as we saw in the last one. They take all the existing, actually they did this in a previous publication. They took all the existing Rubisco sequences and they made a big phylogeny and said, oh, they all divide into different clades actually. And here's the clade that they think is most likely one that existed way back then. And they look at that ancestral sequence and they say, let's make it. And the reason why they want to make it is because they have a hypothesis that in the past, they have this knowledge that Rubiscos, they have when they fix carbon, they have like a bias in them, or they can have a bias in them for certain isotopes of carbon. And those isotopes you can measure in rocks from way back then. And so like there's a thought process that Rubisco from way back then has a particularly balanced way of incorporating isotopes into the carbon back then. But of course, like we don't really have a way of testing. Like the only observation we have for that is like the carbon isotope ratios that we have sampled from the past. And so the way that these authors want to support that hypothesis is by saying, well, if we reconstruct the ancestral Rubisco from that time period, we should expect it to have that. It should have the same carbon isotope preference as predicted by the rock measurements. So yeah, they're doing a computer-based technique, right? A data-driven technique to find a sequence of a protein that then they're going to make to verify a geological observation that you can measure in rocks.
Yeah. Incredible. Like a couple of the things they do, again, like very similar to what we explained before, going back, working back the timeline to find like what the ancestral Rubisco looked like. I think they do test out. So once you create it, you can test out against the current strains of Rubisco. It does less well, which again, it's kind of fun. I mean, yeah.
Yeah, it's hard to say how to verify. Like I think it seems very difficult to verify.
Yeah, because you have to test it in that ancient atmosphere, right? And
and they only have geological records to understand what that ancient atmosphere could be. And so right, it's like they're the same thing. They're sort of like informed by geology, really, in terms of like setting up their experiments. Yeah.
I mean, I would have assumed that they had different versions of this, like ancient Rubisco, because again, we have no idea what the actual one was. They're just trying to approximate it as best they could.
Yeah, because there's probably like it's also silly to think that back then there were just fewer forms, right? Like there may have been lots of diversity back then as well, right? And we're seeing like some successful piece of the diversity. So yeah, that's another tricky part to this. It's like not like things were necessarily less diverse in the ancient times, but this is like one representative from that diversity that could have existed back then.
Yeah, because I mean, I was thinking in terms of like this is their best guess, and I'd have hedged it a bit by having like a few of my other less good guesses kind of in the background to almost like paint the whole picture, just in case like one of the variants is like there is a lot of divergence there that there could be, like in terms of selecting for like slightly heavier carbon versus slightly non-heavier normal carbon.
Yeah, no, I understand what you're saying. And like I also wonder, like again, this is my ignorance of knowing this method of like finding the ancestral sequence, but I'm wondering like they find one ancestral sequence because that like is the most probable one that gave rise to all these extant ones. But is there a way of also getting diversity there? Like is there a bunch of different ancestral sequences that could have resulted in all this diversity?
True.
Because like that might also like sort of cast the net a bit wider to give us a sense of like, you know, like they only have one thing they can test here, but if they had like a bunch, maybe that would make us feel more confident in some ways that like, yeah, like this was happening back then.
Yeah. Because again, it's impossible to like go back in time and get a version of the strain and then have a comparison. So we're just going off of like how good the algorithm is.
Right. But yeah, that's that, and so they do clone this ancestral Rubisco into an algae, and then yeah, they're sort of evaluating it whether or not it stacks up, and they find that it does have the same in a 2% carbon CO2 atmosphere, it does have the same isotope preferences.
And again, that isn't as fun as I was hoping it would be, because I think that would be weird. But I mean, it does confirm all the evidence that they had before, which is
right.
I guess what you'd expect, but what you expect is never as fun as if
it was if it was wacky.
If it was wacky.
Yeah.
If it was like, oh, it doesn't accept carbon, it accepts selenium instead, or like something crazy like that.
Yeah, and I think when I first picked up this paper too, I thought they might have like somehow maybe this was just I think I wasn't thinking straight, but I was like, did they find ancient DNA? I thought maybe they might have found ancient DNA in some ways, but like this is a really long time ago. Like this is not the window in which we can find those things.
Yeah, mosquitoes didn't exist in this era, so therefore they couldn't have
imprisoned some blood from a dinosaur. Okay.
But yeah, this idea of predicting, like finding out the function of ancient genes based on their descendants is very interesting to me. And again, like speaking of dinosaurs, this could, part of me is, a small part of me is always going like, could this science be used to make a dinosaur? Could we get all the bird DNA, work it back to create the?
Yeah.
This is why I don't get grants.
Okay, well let's push forward. We're already at 11, and we have 4 more papers left. This one coming up next is humanization of wildlife gut microbiota in urban environments. Actually, in my mind, a very straightforward paper. Actually, it took a bunch of data that was already collected from gut populations in urban and rural areas in humans, and then also urban and rural areas in coyotes and sparrows, and it merged it with some experimentally collected data of the gut microbiota in these lizards, both in like more urbanized areas and in less urban areas. And then they calculate the relatedness of those gut microbiotas between both the animals in those 2 zones, but also those animals in those 2 zones as compared to humans in those 2 zones. And what they find is that folks that things that live in urban environments are similar, are more similar to each other. Well, not more similar than within species, but they share. There is a signature that's shared in the urban environment that drives some similarity.
Yeah. I find that very interesting. A lot of them are eating our trash, so coyotes I'm thinking of specifically, but then like because I think like humans,
I mean, I think
we kind of underestimate, well, I personally underestimate how much trash I produce as a human being and how that ends up in nature and how that particularly affects urban wildlife.
Yeah. Yeah, I mean, sparrows eat our trash too. I've never seen these lizards. I'm not quite familiar with them, but I also assume that they also eat trash if they're living in urban environments.
Yeah.
Yeah, and like those are the foods that we eat. So of course, like the foods we eat, bacteria probably like to eat them. Like our bacteria also like to eat them. As well, they're contaminated with us, right? Like our trash is also contaminated with our own microbiota, that we're seeding into these lizards or into these different pieces of wildlife. And one of the surprising things that they highlight in their, or maybe not surprising, but an interesting thing that they highlight in their results is that there's like specific indicator species that we associate with like the core human microbiota that are being found in these urban associated wildlife. So yeah, so it almost speaks also to the success of our microbiota, that it can, like with sort of repeated inoculation, our microbiota is able to find new environments to sort of latch onto.
Yeah, I mean, we're infecting other animals with our bacteria, and not only just trash, but we're also like walking around pooping and farting everywhere, and just so yeah, we are like, there are loads of us, so yeah.
That's all I really want to say about this. That yeah, urban environments are having an effect on wildlife microbiota.
Yeah, and talking of microbiota, so this was looking at the gut microbiome of century-old snail specimens that are stable across time and preservation. So this comes from like the Colorado Museum, where they had people going out collecting snails for I think over 100 years as like kind of, it sounds like a student activity, and now they've gone back into that record and found these alcohol like soaked specimens and decided to firstly test whether alcohol preservation is a good way to preserve like snails in a way that is allowing them to get microbiomes. And also see like what this can tell us about how the microbiome of these snails have changed, because snails, they don't move very fast, they stay in one place for a very long time. So the question is like, how do that, so we've had changes in environment in some places, so how does that like, how is that reflected in the microbiome? What they actually found was that the microbiome tends to be quite stable in snails. The only thing that changes is if the snails have been in a different location for a while.
Right.
So they found like diversity across the different locations they sampled, but not much. They also like kind of validated that, okay, it's very good to the way they did the preservation of snails is good. They're happy with that. Although it was kind of grim reading, like how they preserve the snails, because the first thing they anesthetize the snails by drowning them, and
Yes.
they kind of speculate that this loses some of the microbiome because the snails expel a lot of their microbiota when they're being drowned. But
Little
on the other,
last gasp.
so I, but yeah, so this is interesting about like what these snail specimens can tell us about kind of how we can sample microbiomes from different areas, looking at
Yeah.
just as a aspect of the environment. But yeah, that's
Yeah,
very
I find
short
it weird
paper.
that they say that I find it strange that their conclusion was like, it seems good enough. I think good enough is maybe like the best way to think about the
Yeah,
preservation method, because like there were really, there are big differences in the microbial richness between the preserved stuff and the fresh stuff,
yeah,
preserved snails and fresh snails. So like, but you know, it does capture some amount. And so like, I guess the thought is that you could do some sort of analysis. You just can't think that like these are like super accurate snapshots of the past, but
yeah.
they are snapshots of some sort.
They aren't complete, and
Yeah.
at this point, they've been collecting these snails for 100 years. Changing their system now might be a bit of an ask.
Yeah, absolutely. But I think more it's like just value itself, like paying attention to these museum collections from the
Yeah,
microbiota perspective. Like we know, I mean, I definitely have seen, like I watch like some PBS YouTube channels and stuff, and like they talk about like, oh, the great thing about like collecting museums is that you go have all this like this record from the past, and bringing DNA technology into that has then give people genomes from the past, right? So like this is kind of like the next another like innovation on that, right? It's like also the microbiotas from the past could be there. But yeah, of course, with some degradation, so there's limitations to what we can say.
yeah. That brings us to the penultimate paper, which is a volatile from the skin microbiota of flavivirus infected host promotes mosquito attractiveness. So, this like indicates. So they found that this was the first started off by like them testing mice. They had two groups of mice, one infected, one uninfected, and they had like above them they had these like two boxes, and in between them they had like a box of mosquitoes to see which one they'd go to first. And they
Yeah, wait,
found that
roll us. There's a visual of this in
there
like
is a
figure
visual
one.
of this, which is quite fun
I
to
just
look
think it's
at.
funny that the mice are in Erlenmeyer flasks. I don't think they are, but like
Yeah,
maybe.
no, you would not keep mice in Erlenmeyer flasks. That'd be a bad idea. But the idea is that there is there's basically they're wafting the aroma of the mice to see
Yeah.
whether the mosquitoes react to it or not.
hmm
And they find that for Zika virus, mosquitoes are attracted to the smell, and also for dengue fever.
Yeah.
Um,
So
so
something about the viral infection is influencing mice to create nice smells for these mosquitoes.
yeah. So they delve into it and find they kind of took scrapings of the mice. So actually, no, they filtered the aroma through, like and did some mass
Yeah, they did
spectroscopy.
mass spec on the aroma.
Yeah, to figure out what it was, and they narrowed it down to a couple of different compounds that were different between them. Then what they did was this mosquito antenna assay, where they extracted the antenna from the mosquitoes, put them onto electrodes, and then added each of the volatile compounds to see which one caused the antenna to create an electric signal. Um, and they narrowed that down further and found like which ones the mosquitoes were attracted to,
hmm
and then they like looked at then they tested them to see which of these individual compounds that the mosquitoes were interested in would cause them to bite preferentially bite a mouse, and they managed to get it down to one compound which is called what let me remember what it was called acetophenone. Let me look at
Yeah,
my notes quickly.
acetophenone.
Acetophenone, yeah, that's right.
hmm
um and they did further experiments. So firstly, they checked whether humans produce this as well, and
hmm
I think they found that people with Zika virus did produce it.
hmm
Um,
hmm
and then they did things like they smeared human hands with it to see whether it could attract mosquitoes. Yes, it could.
Yes.
Um, and then the question is, what is producing the CETA thing? Because it's not naturally produced by humans, but they figured out, oh, it's produced by bacteria. So what bacteria are producing it? So they did some things like, is it in the gut microbiome? No, we're not farting this out. This is
Yeah.
something on our skin. Um, and based on that, they narrowed it down to a certain set of like members of the skin microbiota, which are like the Bacillus, Brevimunt, Staphylococcus, and things that are like tend to be upregulated. So they looked at my skin and found that those strains were upgraded like 1.5-fold. So that there are a couple of bacteria genera that seem to be more changed in infected mice compared to uninfected mice. And
Yeah.
that those bacteria are known to produce acetophenone.
Yeah, I find this
So
like this is like kind of crazy because, well, just to like try to broaden it for a moment before we dive back in, like Zika and dengue are both transferred by mosquito bites. And
yeah,
so, right, like this is in their evolutionary benefit, like when they're inside of an organism to have it be bitten by a mosquito. And so it's very interesting to think that there's some way in which the infection of it is interacting. So they're sort of following the mechanistic trail right now of like how does this all happen? But like the sort of that end result that like you're more likely to be bitten by a mosquito, or mice are more likely to be bitten by a mosquito if they're infected by these viruses. Yeah, like that's like a really powerful thing for the viral life cycle.
yeah. And the fact they've linked it now into the microbiome. So at this point, they've found the bacteria that produce it, but then they don't understand why do these bacteria suddenly proliferate in mice that are producing this compound. So what is causing that? So in this point, they do a whole transcriptome sequencing to compare the transcript abundance in the skin of virus-infected mice versus virus-uninfected mice, and they find that that a specific gene is downregulated a lot. A gene that produces a resistant molecule, so a resistant-like molecule, which is an antimicrobial protein that's produced naturally by our skin cells. So, and they check
So
that.
it's like a remote immune effect, right? Like it is like you have an infection, you have this immune response. Just so happens that the immune response like also like is modulating another environment in the body that then gives you or increases the type of metabolism that body site has. In this case, the skin. Mm-hmm.
And a fun fact about this protein is that it can be induced by dietary vitamin A derivatives.
Yeah.
So they could administer a vitamin A derivative to virus-infected mice to see whether they're attractive to again, it's got this like entire chain of whether it can restore the expression of that protein and then go back down the line to make mice less attractive to mosquitoes again. And they found that that was the case. That once you were giving mice this vitamin A derivative, which was I think called, I can't remember what it's called, isotretinoin,
Yeah.
and that discouraged mosquitoes from biting the mice, which is like got this massive chain of things going on.
Yeah, it's kind of wild. I mean, of course, the final figure of like just feeding vitamin A, I'm sure they're thinking about this like therapeutic interest. I mean, it's also good to just see they can modulate they could have modulated anyway, in my opinion, the production of this defensin on the skin, right?
Yeah, I fully think that this is like the cheapest way they could do it. They
Yeah,
didn't have to do any, usually you'd have to do a knockout or like a knockdown or something like that. Here they were like, we have you can just do it with the vitamin A derivative.
Yeah, just to like close the loop on that line of logic, right? That it's the defensin that is modulating the phenotype of being bitten. Yeah, just very cool. And again, like this action at a distance is what I find very fascinating about this. Makes me think of like the cordyceps that like takes over the ant brains. I mean, like of course there it's like, oh, it's like affecting the nervous system, and so it's getting to these places that the parasite will be more successful. But like this is also something that in some ways it's changing the biochemistry of the host in order to become more attractive for its particular route of transmission.
Yeah, I mean, there's lots more stuff to delve into, like how are the viruses affecting this specific area of the transcriptome?
Yeah, like what are the viral factors, right? That would be really curious to know.
And secondly, like how many other viruses use this as well? Because it seems to be a signal that the mosquito has evolved to respond to.
Yeah.
So
What about tick-borne disease?
yeah.
Right? Are there
Tick-borne
attractants that? because then we're moving also back into bacteria, right? A lot of tick-borne things are bacterial diseases, and like I like to know if bacteria are controlling the types of things that the types of compounds that we're exuding into the environment to make us more attractive to be bitten by ticks.
And then there's the phages that interact with the bacteria, which also interact with the.
Don't forget about all the layers of life that pile on top of each other.
Layers upon layers. It's viral turducken.
All right, so our final paper for today is the impact of fungal hyphae on growth and dispersal of obligate anaerobic bacteria in aerated habitats. So again, this is like following up. I feel like I'm always, once we do a deep dive on something, I become like sensitized to the stories in the literature that sort of build on that, the central observations of some of the things that we've covered. And this is an extension of that cyborg soil, that cyborg soil episode where we talked about fungal hyphae. They're bigger than bacteria. And so they're able to split up environments. They're able to move longer distances. And yeah, they're really quite important to forming the microenvironment of the soil. And so in this particular experimental setup, they have these like pads of agar, and they have obligate anaerobes, so they can't live except in anaerobic conditions, and they have like air gaps where if they move through this zone, like they're going to go through some oxygenated areas. And they also have a fungus that's able to grow across all those environments. And they're able to show whether they actually do some very in-depth sort of behavioral analysis of the anaerobes as they interact with the fungal spores and the fungal hyphae. Yeah, and ultimately they find that it's dispersal. So yeah, so here's a setup that they do. They show in figure 2 that like this is the fungus is actually using up oxygen, right? It's an aerobe, and so it's using up oxygen. It can create places where there's less oxygen. It also loves to grow in places that there is oxygen. But when they zoom in into the co-culture and they can actually look at sort of these spores of the Clostridia by the fungus, they can see them associating quite closely. They also end up taking like movement measurements, saying that like in the sort of like when they are exposed to like a wet interface along the water film, along the path of the hyphae, like the Clostridia like move or don't move based on the oxygen saturation, right? And that can sort of allow it to disperse from places of higher oxygen to places of lower oxygen where they really want to be and they want to propagate. Oh yeah, there's videos.
Yep, there are videos, so you can see the first funguses, fungal hyphae. You see this like kind of anaerobic bacteria, kind of it isn't able to grow at the moment, but then these hyphae just enter into there, and suddenly now this bacteria can grow.
Yeah, so it's like because the hyphae like they use oxygen, so they can create those anaerobic environments, but like the complexity of this is they're sort of pushing for like even more roles that the hyphae can fill, right? And so like the fact that the hyphae like extend between zones and that the hyphae are filled with or have a water film associated with it allows bacteria to move along them as if they were little highways. Yeah. And I think they do their final model like is a place. I think this is the first thing I described, is that like there's a place where they can have like an air gap, right? Where it's like there is no way to cross the zone of aerobic area, and if the fungal hyphae are associated with spores, those spores can just be moved across that gap where they can germinate once the fungus depletes the oxygen in that area.
So yeah, really like interesting. Again, showing that funguses are just this massive keystone species in the soil microenvironment.
In the soil,
they do so much for the bacterial community.
Yeah, and also I really like the emphasis, like the size, right? Like the being able to link the scale. Like bacteria live in microenvironments, and they're not always able to easily disperse out of those zones. They require other organisms to move around. I mean, that's like even the story. We can even link this back to the urban microbiota, and that we're seeding other animals with our microbiota, right? Like those bacteria become dependent on us in some ways, right, to find their success in these other niches, other opportunities to diversify in the guts of other animals.
Yeah. Completely. We are very much. I mean, I feel like we're fungus are because I feel like fungi they perform so many different functions, and but
well,
yeah,
I think fungi are closer to the scale of microbes, right?
Yeah.
Like they're in the soil environment, like they're that much larger than a single bacterium, but they're also able to move right across these like widely different areas, and that's what gives them such importance in that area, in the soil.
Yeah, that's all great.
Yeah, so that's everything that we have for this week. A bit longer. I think like that we're doing these 2 weeks. We're kind of like getting really into things. Also, we shoegazed a lot into SARS-CoV-2 this at the very beginning. But yeah, we'll be back in 2 weeks where we're gonna try this again and cover the latest microbiology papers that we've seen in the news.
And we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. So science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter at microtwjc or on Mastodon. We both believe that peer review is a process that we can all participate in. So if you've had a good time listening to us ramble on about microbiology today, or if you think you have something to add or found something unclear, just let us know in the comments.
Yeah, it's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content. Goodbye.
Bye-bye.